S&P week. All right. So this is your last content lecture for exam 1 section. That means this time next week you'll be taking exam 1. Okay. Review session on Thursday. Please bring questions. For that, I won't be covering any new material. I'll just be going back over things to help you all understand it better for the exam. Lab today, I'm hoping that we get out early. You know, it's a matter of, you guys are all seniors, juniors, you've done this stuff. So hopefully we'll get in there and crack out the results, and as soon as you have your results, I'm happy to let you go, okay? All right, let's get started. So, we left off talking about some of the highest level processing in the brain, things like facial recognition. And so we're going to start with finishing this program. All right, so what do y'all think about that? Interesting? It's pretty complicated. I mean, we can look at, just like they did in that study, right? And we can show people faces or objects, right? And look at how their brains are recognizing those things in a scanner. And it looks like these two separate areas are responsible for two separate really complex tasks. So the fusiform area, responsible for processing and measuring faces, and this other area called the parahippocampal place area, or PPA, lights up near the hippocampus for non-facial recognition objects. Right? But if we go in and we look at the brains of the people who are born without the ability to recognize faces, it doesn't look like there's anything wrong with their fusiform area, right? That was their prediction. But when you go in and look, it looks like it's just fine. So there must be something else missing there, right? It's much more complex than just this area does this and this area does that. All right, so here's another study where they overlapped faces and objects, and then one of these objects would slightly move, and that's the one they wanted you to attend to, right? So again, the important thing for a lot of these tests is that your brain is seeing, sensation-wise, exactly the same thing, right? But your top-down influence of attention is causing you to focus on one thing or the other. And when they do this study, and they look at activity in the fusiform area, when people are supposed to attend, when people down here attend to the thing that's moving and the face was moving, fusiform area high, right? When they're supposed to attend to the thing that's not moving, that's static, and the face was static here, the house was moving, again, fusiform area is high there. And the same thing for the parahippocampal place area, when you're supposed to focus on the house instead of the face, we see the opposite effect, right? So activity goes up in the parahippocampal place area and goes down in the fusiform area. So pretty good evidence of the fact that these two areas in most people are working to process complex images of faces and fusiform parahippocampal place area for objects. Another test here, again, nothing's changing. They have you fixate on this dot, and then they say, okay, we're going to put a red or a green oval. We want you to attend to the thing. We want you to focus your attention, even though you're not moving your eyes, right? But you're paying attention to the thing that's highlighted in red. And when the face is highlighted in the thing we pay attention to, we get more activity than when you're supposed to pay attention to the house. That activity goes down. So again, nothing changing sensation-wise. there's still an image of the face and the house being projected into your V1, into your brain, right? But whether or not you're focusing on that changes brain activity so that it goes up when you're focusing on the thing, the face and the fusiform area. Does that make sense? So a lot of what I want to talk about today is that relationship between neural activity and perception. And in general, more neural activity means more perception, right? When you're attending to the face, it generates more neural activity. All right. Yes, activity of FFX increase when you pay attention to the face. Okay, so this sort of leads us to, if we have all these top-down influences on our perception, when do we really see something, right? If these incoming sensations can all be the same, but our brain area can change based on attention, like when is it that we truly perceive something? And so in this study, they present a face for a very, very short period of time, 50 milliseconds. And right after they present the face, they present a masking stimulus. So it's a very difficult task. And then they ask people, did you see a face? Did you see another object? Did you see just a random texture? Right? And at the same time they're doing EEG, this is now not fMRI, which remember fMRI is not really good at very small time resolution. But EEG is very good at that, right? And so when they look at the activity in the fusiform area, in the seconds right after this perception happens, right? Actually, I'm sorry, this is fMRI, not EEG. That's why it's 5 seconds, because it's not great time resolution. But this is only presented for 50 milliseconds, and then the mask. If they weren't shown a face, if it was just a random texture here, not a face, right, that's the blue level of activity. If they were shown a face and they were able to say, yes, I saw a face, they have the most activity in the fusiform. But what's interesting is if they were shown a face and they said, yeah, I didn't see a face on that trial, right? They incorrectly said that. It was sort of this intermediate level of activity, right? So down here they don't see a face, there's no activity. But here they see a face but don't recognize that they're seeing a face because they haven't crossed some threshold of activity that allows us to perceive a face, right? If you don't generate enough activity in the fusiform gyrus, then you miss seeing the thing that actually was projected on there, right? So again, figuring out these relationships between brain activity and perception. All right, so we're going to talk about another experiment. This is done with a monkey, using something called binocular rivalry. So remember, half of each eye crosses over and matches up with the other half so that we get consolidation of one image, right? So when you look up at me and see Dr. Pittman, right, you don't see two of me because I'm hitting both of your eyes, but that signal's getting consolidated. We can confuse the brain by putting a different image on each eye. So here, in this case, this is called binocular rivalry. Why is that involved there? Because that's a term that you need to know, right? Binocular rivalry, right? Which means I'm showing something different on each eye, and so those images are producing a rivalry. They're competing with each other for your perception. And in this case, don't worry about yours. So in this case, this monkey's getting a starburst on one eye, and a butterfly on the other eye. Okay, And so when that happens, your brain's trying to consolidate two different images. So take the mirror like this, okay? Put it on your nose, and then tilt it so one eye is seeing whatever's in the mirror, and the other eye is looking at me. What are you seeing? Especially if you can put it on. Right? So it's like your brain is trying to focus on two different things, two different images, and one's winning and one is losing. And it kind of flip-flops back and forth, right? Between seeing the thing that's in the mirror, seeing the thing that's right in front of you, right? It's like trying to blend it together, right? Okay, so this is what we're doing to this poor monkey. But at the same time, the reason we're using a monkey is because monkeys will let us record from their brain. So we're recording from, we find a neuron in IT, okay? Just pass it down. We find a neuron in IT that responds to the butterfly. So when the monkey sees the butterfly, that column gets really active. And so the monkey's trained, hey, if you see the butterfly, if you see the starburst, push the left lever. If you're perceiving the butterfly, push the right lever. So just like your brain was flip-flopping back and forth between what you were seeing in the mirror and what you were seeing in front of you, same thing's happening for this monkey. And he's going in. Starburst, oh no, now I see the butterfly. Okay, no, now I see the starburst. And what they're able to do is then correlate that with the activity of that IT, inferior temporal neuron, that responds to seeing the butterfly. And every time that column in IT gets really active, it's the same time that the monkey's indicating I'm perceiving the butterfly. It's like a direct relationship between the amount of activity that's happening in IT and which of those two images we're perceiving. Which of those two images we're perceiving. All right. So now we're going to talk about focus, vision, and attention. And the role of attention. I've already talked a little bit about the role that attention can increase brain activity. And when we increase brain activity, we see better perceptions. All right. And so again, the big question here that we're trying to ask is to understand this relationship between sensations that are coming in and what we see or perceive. All right, so remember at the very beginning of class I told you that I do this class for fifth graders, and we'll see how good you are versus they are. Okay, so if you've done this before, let everybody else do it, and then we can talk about it afterwards. But I'm going to show you a video of two groups of 3 college students. One group of 3 college students is wearing all black. One group is wearing white t-shirts. Each, you can think about them as like the black t-shirt team and the white t-shirt team. And each of them has a basketball that they're passing to each other. Your job is to count the number of times that the team with the white shirts passes the basketball without getting interfered with by the team with the black t-shirts passing their basketball. Okay? And we're going to do this twice. So just when you're done, just write down the number of times that the white team passed. So here's white shirt, white shirt, white shirt. The rest are in the black. And we're going to make it a little difficult for you by making it a little transparent. All right, everybody ready? Focused attention on the team with the white t-shirts. All right, everybody's got a number? Yeah, write it down, okay? All right, I'm gonna make it a little bit easier. We'll do it without the transparency. So, ball is starting right here in his hand, okay? Ready? One more time. Here we go. All right, we got a number. What was your number from the first one? 16, around there? Well, I know. What about the second one? Right around 16, 17? Right? All right, did you notice anything unusual in those clips? Hayden's saying yes, what Hayden?
The gorilla.
The gorilla. What? How many people saw a gorilla? How many people did not see the gorilla? How many people saw a gorilla in the first clip? All right. Yeah. So if you've already seen it, right, remember Heracles said you can never step in the same river twice, right? So if you've seen this before, right, then it kind of ruins it. But the first time you saw it, you probably, the idea is you're so focused on ignoring things that are dark colored, right, that you don't see the monkey walk in the middle of the rim. And some of you actually missed that, right? Okay, because you were so focused on counting and not being interfered with and other things, right? So, whoops. Here we go. All right, let me show you this video. All right, anything unusual there?
He changed shirts.
Change shirts. Isabel?
He looks kind of different. His face.
Yeah, it's two different people. Right? And yeah, they're both wearing glasses and they both have blonde hair, but two different people, right? And you are expecting that, right? That doesn't normally happen when you're watching a clip and someone gets up and walks out a door and then someone walks out into a hallway. You're expecting that to be the same person, right? There's no reason to think that it's going to switch. And so your brain kind of glosses over pretty big details, right? If you're not paying really close attention. All right, so again, here is another test where they're showing somebody two arms, and the horizontal and the vertical arm, one of them is longer than the other. And the goal is for the person to correctly identify which arm is longer. And while they're looking at this, they will sometimes put an object in their visual field, like this triangle, and then ask them, hey, did you see one of these objects? And the idea is that if you're paying really close attention to figuring out that this one's slightly longer than this one, you miss the fact that they flash a triangle or a square or a circle into your field of vision, because that's not what you're focused on. Does the triangle stimulate your V1? Yes, but it's at a level that you're not paying attention to, and so you don't perceive it higher up in the cortex, right? That signal's still coming into your retina, still going to V1, but it's top-down influences in V1 that are sort of picking and choosing which columns to highlight as your perception, right? And this is what we call inattentional blindness. Because you're not paying attention to something, you're blind to it, even though you had an incoming sensation for that thing. You had an incoming sensation for that. All right, yes?
So are these really like conscious or more conscious attention? Because we talked about in confidence a little bit about how you're still processing everything and things like the propaganda that you're being primed. So is this more like you're more actively focused versus if you're not paying attention, but can still be influenced?
No, here we think you're actually not you're blind. You're not perceiving the thing because your attention is focused elsewhere, right? So you are seeing it in terms of your eyes. You're seeing that signal. It is going to V1, but it's not getting consciously recognized out of V1 into these processing streams because of the top-down influences of attention on other things. Okay, on other things. Okay.
So is that kind of tying back to like when you're driving, you kind of zone out? So
Sure.
like you're still seeing it, but it's not enough to really like focus on it. So you're like, oh, I don't even know the last 5 minutes that I just drove. Something like that.
Something like that.
Okay.
Yeah. And that may also involve the level of the thalamus sort of filtering things to there, maybe even the signals being dampened coming into V1 because you're drowsy or you're tired, your arousal's down.
So does that information just completely stop at people?
Yeah, if you're blind to it, you don't actually see it, right? Perceive it consciously. Okay, all right, so we're gonna do another test. So look up here. I'm gonna show you a whole series of letters. They're gonna flash really fast. Mixed into the letters or numbers, your job is to see the numbers, right? The letters are the distractor. You wanna perceive the numbers. All right, it's gonna be real quick. We're gonna do it like 4 times. So look up here, ready, set, go. Write down number if you saw number. Okay, let's do it again. Right here, look here, we'll do, this is a different test. Ready, set, go. Go. Numbers. Right, okay, one more, or 2 more. Right here, ready, set, go. All right, last one. Looking right here, ready, set, go. All right, so sometimes even if we're really trying to pay attention to something, the fact that we're paying attention to it can cause us to miss other information, right? So, and this is called an attentional blank. It's almost as if paying attention to something causes us to blink and miss some information, right? So in the first one, how many people got 2? Okay, how many people got 7? 2 and 7. Okay, only 2 of you. Okay. So what happened was, I'm flashing these numbers and 2 comes up, and it's like your brain goes, 2, remember 2, and then it goes back to looking for numbers, but by that time, there's only one letter in between the 2 and the 7, and so you miss the 7. It's like your brain blinks while it remembers the 2 and it doesn't catch the 7. In the second test, how many people got 2 and 7? Only 1 person. Okay. Well, usually everybody sees the 2 and the 7 because they're spread out further apart. So you have plenty of time after you see the 2 to go back to the test and see the 7. My demonstrations don't always work. But normally that's what happens. But here's where it gets interesting. What did you see in the third trial? A 2 and a 4. Okay. In the second trial, there was a 2 and then a red 4. Right? What's interesting is that this is at exactly the same position here, and you miss the 7. What's the difference? Stimulus salience, right? So now, because the 4 is red, you perceive it. If it had been black in color, you would have missed it, just like you missed the 7 here. Right? So what does that tell you about your visual system? It likes contrast. And did you have a sensation for this 7 here that nobody perceived? You did. Because when we change this to be red, all of a sudden the fact that the color's different pulls it out of that blank and says, hey, that was different. and you all of a sudden perceive it. You can't perceive it if it never came in, right? How many people got the 2 and the 4 in the last one? Again, because they're 2 different colors, even though they're in exactly the same positions, right? We see it. So that's just showing you that it's about 500 milliseconds, about half a second, is how long this attentional blink usually lasts. When you're trying to see something and you're like, oh, I recognize that, remember it, and then go back to the task, it takes about half a second to do that. But you're still seeing things in that half second. And if what happens in that half second, right, is important, if it changes color, right, then we can pull it out of that blink and perceive it. Right? So this is sort of demonstrating to you that all of this stuff is still coming into your V1, but it's like how you pick and choose what you pay attention to, right, affects what you're perceiving and being able to see. Okay, so let's look at this other clip. Yes, sir.
On one of them, like the one where the 2 and 7 were spaced out, I saw 2 and 3. So is it like?
Yeah, you probably saw a letter that looked like 3. You're trying to see numbers, and so something that you probably saw E or something like that.
And just like filled in.
Yeah.
It's been a long time since I've seen you. Yeah, it's great to see you, Andrea. So how did you get here? I took the subway from Middleton and it took only about half an hour.
Really? I drove from Gresham and it took 45 minutes.
All right, public transportation. So why did you call me here for this mysterious meeting? I'm planning a surprise party for Jerome, and I need your help to keep him away from the house.
That's great. I'll do anything you need.
Good.
I hate surprise parties, but only when I'm moving there. Otherwise, they're great.
Very good.
All right, did you notice anything unusual? Yes, Mary? Mark.
Lights were changing colors, and what was on them was changing, like there was food on one clip, and then there was trash, and then there was switch.
Did anyone see that? Anyone see that? Some did, some did not, right? Right? So we call this change blindness. I mean, when have you ever been at Burwell and you look down and all of a sudden the food that was on your plate is on your friend's plate and your friend's plate that was empty is in front of you? And then you look back up and then you look down and it's switched back. Has that ever happened to you ever in your life? No, that doesn't happen in the real world, right? And so your brain doesn't expect that to happen. And so it sort of glosses over things. So if we look here, right? She's got the red plate, paper cup, the trash is on her plate.
Hey, it's great to see you. Yeah, it's been a long time. Yeah. So how did you get here? Oh, I took the subway from.
She has a scarf on right now, the woman in red. Whoops, what happened? That's not the same clip. Go back to the first
Hi, Savina. Hi.
one. Sorry.
It's been a long time since I've seen you. Yeah.
She does, right? She has the food, red plate. Now her scarf is gone. How many people notice that? Right?
Really? I drove from Gresham and it took 45 minutes.
Now the scarf is back.
So why do you
Now the
call me
plates
for
are
this
white.
mysterious meeting?
Right? Now the plates are
I'm
red.
for Jerome, and I need your help to keep him away from the house.
That's great. I'll do anything you
Now
need.
the trash is on her plate and the paper cups over there and the plastic cups over
Otherwise, they're
here.
great.
Very good.
So lots of things are changing in there, but unless you're specifically looking for abnormality, right? If you're just watching this thing, trying to perceive it, our brain glosses over those things, which is what's called change blindness. Look at one or two more examples. We won't take all of these. Right? So you focus on that white center part. Right? You see what's changing? You want to see the change? Okay, so you have to, if you look around, instead of focusing right in the center, right, scan around the picture. You see this building is coming and going over here? But only if you look right there at that part are you able to see that, right? So again, if you focus on that fixation point, right, it becomes difficult to see that unless you scan around the picture. When you scan around the picture, then you can see that the chimney is disappearing into here. What about this one? You don't see the change? Nobody saw the change? All right, let's try it again. Don't see the change? No. All right, so again, you're scared, you're trying to see a change, right? But unless you look at this rock down here in the lower corner, right? Unless you're happening to stare right there, right? As that fades out of the picture, then you don't see it disappear. You have to be looking right at it, right? Even though this is all coming in, but you're not able to perceive it. Again, that's what we're trying to get to. So I'll show you one more. All right, anyone see the change? No? Okay, look at this rock right here. So that rock's not there in the beginning, right? And it slowly fades into the picture, right? So again, even though you're actively scanning, you're like, something's gonna change, I need to find it. It's not enough to simply have information coming in. We have to be paying attention to that information in order to see that. Okay, so selective attention is necessary for us to detect these changes in our visual environment. And while stimulus salience can play a role in helping us detect changes, right? It's not always sufficient, right? Because in the flicker test and in the gradual change, you are having something that's changing, but unless you happen to put it in your field of vision, to your phobia, and really process that, you're not able to detect it even though it's flashing on and off, all right? Okay. And again, we have this knowledge bias that like rocks don't disappear from rivers, right? That's not something that should happen, right? Plates don't change colors, so my brain glosses over that, right? And that's part of these top-down influences that we'll talk a lot more about in our next exam section. We're just starting to scratch the surface a little bit here, but I think it's important that we know that sensations don't equal perceptions. Sensations don't equal perceptions. There's all these influences that influence that. All right. And the fact that the difference between perceiving something and not perceiving something can often be the amount of neural activity that that thing is generating. And if it's not generating sufficient neural activity, then our brain doesn't pay attention or perceive that thing, even though it's coming in through our visual pathway. All right. So again, here is some experiments with some monkeys. What is before code in the brain? Is that part of the brain too? Color coding, right? So here we have two stimuli, a blue and a red rectangle. And again, in all these things, we're having the monkey fixate on a point so that these things are in a field of vision, a receptive field, and they're not moving out of that receptive field, right? And so here, the ring is a signal to pay attention to a particular stimulus. And so if it's the preferred stimulus, then that means that the monkey, if it presses a lever, can get rewarded. If it's the non-preferred stimulus, they can't get rewarded if they press the lever, right? That's what makes it preferred and non-preferred. Here, when it's the preferred stimulus, look at the activity that's happening in V4. When it's the non-preferred stimulus, that activity goes down. The important thing here is that nothing has changed about the color stimulation happening in V4 in terms of bottom-up signaling. Nothing has changed, right? So you would think that bottom-up, the blue and the red would be stimulating V4 the same. But when it has meaning, we upregulate, we turn up that signal to make it bigger in V4 to help us perceive it better. And when it's something we're not paying attention to, that signal gets dampened. Again, here's another experiment where the monkey is fixating here and this little star is getting brighter and dimmer. So again, we're just making sure that this receptive, that's what RF is, receptive field, that this receptive field is staying the same by having the monkey fixate on this point, and then we're making this brighter and dimmer, and we measure the brain activity during this. Then we train the monkey and we say, hey, guess what? Every time it gets brighter, if you press the lever, we'll give you some fruit juice. All you have to do is press the lever when it's brighter. So again, the monkey's fixating, same exact stimulus, same exact bottom-up signaling, but now look at the amount of brain activity that starburst changing intensity is generating because it's meaningful, because we gave meaning and the monkey wants to pay attention to that. So again, meaning, attention, changes, it influences the bottom-up signaling. And when it turns it up, we perceive it better. Better perception, more brain activity. This is in humans, looking at their occipital cortex in an fMRI machine. And again, they're fixating in the center, and what's coming in is a green, this is what they're seeing on the screen, this is their brain activity superimposed on it, but they're seeing a green diamond and a blue circle. And over here, they're seeing exactly the same thing, bottom-up signaling, green diamond, blue circle. The only difference is they ask people to focus on a different stimulus. And when they do, when they tell them to focus on the green diamond, this is the pattern of brain activity that's happening in their visual cortex. And when we tell them to focus on the blue circle, a different pattern of brain activity happens, even though there's no difference in the bottom-up signal. There's zero difference in the bottom-up signal. All right? So our perception is strongly influenced by how we are treating bottom-up signal. We can turn it up and down. Okay. All right, so if you take anything away from today's lecture, write down slide 33. I need to understand what this slide is saying, right? These are 3 examples, right? So over here we have a tuning curve for orientation columns in V1, right? So this neuron that we're recording from would respond the most to a vertical stimulus and drop off as we get less vertical and more horizontal. Good? Everybody understands that? All right. When we make that more important, more salient, or you need to pay attention to recognizing that, the brain activity increases for that thing. Just like we saw with the brain activity increasing in V4 for the monkey when the blue was preferred and the red was not, right? Top-down influences can turn up the signal, attention, so that now that column is responding more, and we're seeing it better, we're perceiving it better. Right? We've also seen examples, remember the weird paperclip shape that the monkey didn't recognize, but once we trained the monkey, it could discriminate between very different angles of that paperclip shape and other paperclip shapes. And what happened to the tuning curve? The tuning curve got more narrow, right? And as this gets more selective, more narrow, it allows us to better discriminate between, is it this angle or is it this angle? So experience and practice can sharpen our tuning curves and make them smaller, and when they're smaller, they're better able to discriminate between stimuli. So as the monkey learned to tell the difference between those paperclip shapes, right, it was encoding and sharpening an orientation curve for its preferred target. And then lastly, we saw how adaptation, when I had those bars that were slanted in different lines and you adapted, and then I put straight bars up and all of a sudden they were bent the other way and then they had to come back up, right? Because we can shift that tuning curve based on adaptation. Unfortunately, that change goes away really quickly after when we're adapted, so that we can shift back to our normal vision. But again, there's ways in which, through experience and what we're seeing and attention, that we manipulate, that our brain is manipulating the basic firing patterns in V1, the basic firing patterns in V1. And that alters our perceptions in complex ways. Okay, all right. Okay. So, just cognitive scientist at Duke, Stephen Mitroff, that wanted to know, what if I can induce blindness where you say you don't see something, but are you still, you say you don't perceive something, are you still seeing that thing from bottom-up signal? And so what he uses is this task. It's a little bit difficult to do in here, called motion-induced blindness, which is where the movement of a large grid, when you're fixating on a point, can cause, that movement can act as sort of like cover up or make you blind to a stimulus in the periphery. So I'm going to show you this. It's hard to do in here, but focus right on that fixation point and don't move your eyes, okay? And I want you to notice what happens to the yellow dot as you're fixating on the white dot. So just fixate on the yellow, on the white dot. Anything happening to the yellow dot? Is it disappearing for some of you and then maybe reappearing?
Yes.
And that yellow dot is not going anywhere, right? That's the motion-induced blindness. The motion of that grid causes interference, and and sometimes then you don't perceive the yellow dot. This one may work a little bit better. So again, just fixate. Fixate on the flashing green dot. Just stare at the flashing green dot. Does anyone have motion-induced blindness? One or more of those dots goes away, even though it's not changing at all in that thing. So here's the question. Okay, great. Super cool illusion, whatever. Right? So here's the question that Mitroff wanted to ask. He puts people in the lab, does this thing on the computer screen, and just like the monkey, he has them push a button anytime the dot goes away. So he knows when they're in motion-induced blindness. They're saying, hey, the yellow dot went away by pushing them down the lever, right? So while they're saying, I can't perceive the yellow dot, he then actually removes it while they're in the motion-induced blindness, so it's no longer there. And the question is, do they perceive that? They're saying, I don't see it. I don't perceive the yellow dot. And so if it's invisible, right, there should be no perception of change. However, and this really is difficult to do, he did another thing where he had this is probably easier to understand than yellow line. So when they push the lever and say I'm in motion-induced blindness, he rotates the line, right? And what happens is the movement of that, even though they're saying I don't see it, just like the red 4 that flashed after the 2, the change in stimulus salience yanks it out of that motion-induced blindness and people perceive the change. And when the circle goes away, they perceive like a little blip of the circle right as he removes it. So they are, at some level, their brain is reading that, even though the movement of the grid is suppressing that activity out of your perceptual awareness. The problem with doing this with you guys is you have to actually be in the motion-induced blindness when the change happens. I can't make it happen when you're telling me that you're in the motion-induced blindness like he can. But this is his example. So he rotates that and then it goes away. And people, even when they're saying, I don't see it, as soon as he starts to move it, it comes into their perception. So subjects can see the change. So this is telling us that bottom-up signaling is still coming in, and the blindness is from top-down influences of that motion, the illusion that that thing is moving over and covering up that stimulus so that we no longer perceive. All right. We're getting there, people. We're getting there. So when you look at this image down here, what are your perceptions? This is called phenomenological descriptions. What do you see?
There's mostly green.
Mostly green? Okay. What if I tell you attend to the horizontal lines? What do you see? Do you see a pattern? All right, they're all kind of making a diagonal here if you tend to just the horizontal lines. Right? What if I say attend to the blue lines? Okay, so now you're seeing kind of a horizontal diagonal line here. Right? What if I say attend to the red lines? Right? You're seeing like an oval shape in there. So again, giving meaning to what, when you first looked at that, you're like, there's a bunch of lines on there. Some of them are green, some of them are red, some of them are blue. Right? But as we attend to things, our brain wants to group things together and make sense, find patterns that make sense of things. Right? Find patterns that make sense of things. And so this leads, whoops, did that too quick. This leads us to the binding problem. How does the brain do this? How does it bind things together? Right? So if you look up at the screen, what do you see up here? A blue car, right? So, and it went across the screen really pretty fast, right? So it's actually a blue Corvette, right? So how is that blue Corvette represented? What happened in your visual cortex when that flashed on the screen? What parts of your visual cortex were activated? V4 for what reason? Because it's blue, right? What else? You didn't say a blue blob went across the screen. PPA, right? Parahippocampal place area. Because it's an object, you recognize it as a car. But before we can get to PPA, what has to be activated? V1. Like all these little donut receptive fields coming in, right, are causing columns to fire, and then those columns are being reassembled, not into angles of lines and spatial frequency contrast, but into objects. And so from V1 and it goes to V2 and V3, which are doing form, and V4, which is doing color. And then we're not just doing forms of like there was a round wheel and a rectangle shape of a car, but then we're taking those and assembling them in IT through that ventral pathway, right? Until we get to the level of PPA where we're recognizing the car and maybe even what type of car it is, right? So think about that. As this goes across the screen, you're binding all of those stimuli together. Even though you have at least like this many areas of your brain and movement. We had MT also being activated, right? IT, I didn't even list PPA up there. So all of these things are happening pretty rapidly in your brain, and your brain is saying, this is one object, I'm binding it all together. How does your brain do that? And do it really, really quickly with no lag, right? This is the binding problem. How do we take areas of your brain that are spread out from your occipital lobe all the way up into your temporal lobe and cause it to be one perception very quickly? Very quickly. Right? So you perceive that as a blue car, blue Corvette, not as wheels and doors and windshields all separate. Right? So how we combine features that are coded in different places in our brain is the binding problem. Right? And it turns out that synchronous activity, neurons that are firing at the same time, become bound together, become that image. So here in this particular study, we're showing a grid of just spatial frequency bars, white, dark bars, and these bars are moving upward, right? So they're just rotating one after another like a bell, just scrolling up, right? And we look at the brain activity that's happening with an EEG in a human, right? So now, this is a pattern of brain activity we get for these bars moving. Okay? Now, the next stimulus is exactly the same, except we have disconnected the bars in the middle. But they're still moving, still same spatial frequency, moving at the same speed, except now there's just a line in the middle. Right? And what happens is that this brain activity gets less synchronous. Because we're not binding this as one object, we're now seeing two tracks that are moving up. And that causes less synchrony in the brain, because we're now binding two things instead of just one thing, more synchrony. Does that make sense? And if we really want to make those two things different, we make one move up and one move downward. And when we do that, we cancel out that synchrony that was happening here, because now we're perceiving two very different stimuli, one grid moving up and one grid moving down. Now, there's another part of our brain that's very synchronous to recognize these two objects, right? That was not synchronous when it was a single object. So don't think about it that way. We're just happening to measure the part of the brain that's very synchronous for a single object moving up. Yes?
So what would be a good way to define like the number of spikes in the graph? On the y-axis.
It's not necessarily the number, but it's the on-off synchronous firing pattern, and it's the size of that synchrony right? The difference between the on and off. For this single image that helps us bind that image together as a single one thing, and as we make it two things or more complex, that synchrony decreases. All right. I'm going to listen to this researcher. He's going to talk about the binding problem, and he's going to use binocular rivalry again. They're going to put a different object on the left and the right eye to make the brain compete. So how does the brain deal with binding that?
So a feature of color vision that's often not appreciated is that color is as difficult to interpret as language. So we think of color as being in light. Color is not in light. It's absolutely wrong. Color is in the brain. It's a construction, just as the meaning of words are constructed. And without the neural processes of the brain, you wouldn't be able to understand colors of objects any more than you can understand a language that you hear but don't know. So what we study are the neural processes of the brain and the eye that allow us to understand color in the natural world. Another aspect of color vision that we often don't appreciate is that the different features of an object, for example, its shape, its color, its motion, its distance from us, those can be represented in different parts of the brain. Yet if one sees a basketball coming towards him, we see that ball as having a particular color and shape and velocity and location. The knitting together, or what I'll call neural gluing, of all those different features so that we perceive a unified object with its color and shape and so on, is a very complex process of the brain that's not well understood. And that's what our research focused on. The technique we use is called binocular rivalry, which pits the signals from the left eye against the signals from the right eye. And the brain has difficulty integrating those signals. When they're different enough from the two eyes, the brain has to somehow resolve what's really out in the world when each eye is not sending signals of the same object. And we take advantage of that feature of the brain by putting a vertically oriented set of stripes that are green in the left eye and a horizontally oriented set of stripes that are red in the right eye, like this. And the brain cannot deal with right eye horizontal and left eye vertical at the same time. It can't mix those together or fuse them in a way that makes any sense. So what the brain does is suppress one or the other. So one sees only vertical or only horizontal. So what we found in our study is that we could develop a technique that though the horizontal pattern was suppressed, the red color of it actually continued onto the brain. It wasn't suppressed. And it glues itself onto parts of the pattern that's vertical in the other eye. And that proves the idea of neural binding or neural gluing, where the color has to be connected to the object in an active neural process. To us, it seems automatic. Every basketball has a color. Every shirt has a color. The brain actually is putting the color and the object shapes together.
All right, so basically what we're saying here is that we have these separate areas of the brain, like V4 that's dealing with color, right? But at the same time, the brain's job is to bind what it sees together into recognizable objects, right? And the way it does that, even though motion's happening over here in MT and color's happening over here in V4,
between the ears and the inner witness is watching this wonderful show, and it's all in color and it's complete in some wonderful sense. But then when you do the physiology and you study perception, you realize, no, in fact, you have a very limited take. You're only taking sips from that fire hose of information that's coming in, a little bit from vision, a little bit from hearing. And there's in fact this competition going on, tug of war between different senses, between different interpretations of what you're seeing. And all of this competition resolves itself in fullness of time and pretty darn quick to produce the behavior that we're capable of and the reflection we're capable of. It seems, though, as if it all comes together in some place for enjoyment in the middle. And that's just an illusion. There's no place in the brain where it all comes together for enjoyment and for witnessing by an inner witness. So we have to take all that work that inner witness was going to do, and we have to break it up in little bits and distribute it around in the brain in the time and space that's available. And no one of those little bits is going to be conscious. And yet the sum of all of that work that the inner witness is going to do, that's what consciousness is.
Well, we have this illusion of unity, a binding, that everything flows together.
We have that illusion, and it is an illusion, because in fact, there's lots of things going on at once. They're not all that coherent. And so our brains are very good at creating the sorts of simplifications that make its own job easier. And so, yes, consciousness is the region of the brain for the brain, by the brain, if you like. And if that seems to be self-contradictory, then reconsider it, because it doesn't have to be.
So essentially what he's saying is that our conscious perception, right, there isn't a single place that's happening. So PPA doesn't have a blue Corvette neuron that fires. What it does is it recognizes the pattern of V2, V3, V4, MT, IT. It recognizes all of that activity that's happening in these different places as coming together to be this thing I have as a memory of what a blue Corvette is. Right? And if I change the color of the Corvette to red, right, you're still perceiving it as a Corvette. It just now has a different color laid on it. But because that pattern was similar enough, the only thing different was V4, right? The PPA still recognizes it as the same thing. Does that make sense? I know it's kind of philosophical and a little bit abstract, but this is why we're studying these senses, is to understand how the brain is doing some of this really complex stuff. Right? And so that wraps up everything for exam 1. Okay. We will dive deeper into top-down influences in exam 2. That's what exam 2 is going to be about. We're going to look at some more complex processing things like color. How does color get turned into color in our brain? How does motion get processed? Those sorts of things is where we're going to move into in exam 2. But for now, mostly bottom-up signaling, but recognizing bottom-up signaling is not the end-all, be-all. We manipulate that through top-down